Hello, I'm Sean, and in this video we're going to explore the similarities and differences between cell-bound and secreted molecular signals in the context of map formation. The mechanisms of secreted molecular signals are actually fairly well understood. These signals are often secreted by a group of cells, and the result of these secretions can take many forms, such as an isolated gradient or a countergradient. Once a cell with an appropriate receptor receives these secreted signals, intracellular signaling cascades often occur, leading to some sort of change such as differential gene transcription or cellular migration. However, what is most notable about secreted molecular signals is their apparent one-sidedness. The only elicited change occurs within cells that receive the signal, which is a phenomenon described as forward signaling. It was shown that cell-bound molecular signals differentiated themselves by their ability to utilize both forward and reverse signaling. With reverse signaling, cell change was no longer isolated to the cell receiving the signal. Instead, the signal-presenting cell could also have important intracellular changes elicited. Moreover, cell-bound molecular signals differentiate themselves from their secreted counterparts by their ability to produce a tighter localization of signals and their highly specific control over the modulation of cellular response. Since cell-bound molecular signals require membrane anchorage via a direct cell-to-cell contact mechanism, cell-bound molecular signals can be restricted to highly specific regions. Additionally, the reverse signaling abilities of cell-bound molecular signaling mechanisms allow for dual regulation of responsiveness in not only the signal-receiving cell, but also the signal-presenting cell. For example, this regulation could manifest itself in increasing responsiveness to guidance factors by increasing receptor expression in both cells. For these reasons, the reverse signaling associated with cell-bound molecular signals has been shown to be pivotal in many development processes such as axon guidance and blood vessel formation, among many others. In order to get a better understanding of the differences between secreted and cell-bound molecular signals, we will look at a couple examples from the primary sensory map formation in olfaction, vision, the primary somatosensory area, and audition. Out of all the primary sensory map formation modalities, the development of the olfactory sensory map is one of the most well understood. The olfactory map depends on linking groups of olfactory sensory neurons in the olfactory epithelium to a specific glomerulus in the olfactory bulb. Olfactory sensory neurons are distinguished by their odorant receptors. Each neuron will only have a single type of odorant receptor, and all the neurons with the same type of odorant receptor will converge on the same singular glomerulus in the olfactory bulb. Additionally, the dispersal of specific olfactory sensory neuron types in the epithelium will be random, but only within segregated zones. What we're particularly interested in is how these olfactory sensory neurons are able to map to a corresponding glomerulus bulb based on its odorant receptor type. Each odorant receptor type will lead to a graded adenylate cyclase activation, which in turn results in a differential expression of plexin A1 and neuropilin-1, both of which are guidance receptors. Plexin A1 and neuropilin-1 utilize secreted molecular signaling to help guide and create a topographic map with anterior-posterior patterning. Olfactory sensory neurons additionally contain ROBO, which is a receptor that is able to respond to gradients of slit for dorsal ventral patterning. These two patterning mechanisms both utilize secreted molecular signaling in order to create a coarse yet robust 2D topographic mapping in the olfactory system. However, the finer details of olfactory sensory neuron axon guidance are determined through cell-bound molecular signaling. In order to segregate glomeruli, ephrin, a cell-bound ligand, is utilized in conjunction with its corresponding EPH receptor to promote repulsion between neighboring glomeruli. It is also prevalent in determining ventral regions within a glomerulus. In order to promote cohesion within a glomerulus, the transmembrane cohesion proteins Chiral 2 and 3 are utilized. The second most well-understood map formation example is the development of the retinotopic map in vision. It was Roger Sperry's experiments on frogs that truly set the foundation for a deeper understanding of retinotopic mapping. When Sperry rotated a frog eye and allowed connections between the retina and tectum to be reformed, it was noticed that there still existed a mismapping of the visual field, indicating that neurons were guided by cues to recreate their original connections. Although the molecular guidance cues that allow retinal ganglion cells to exit the retina and project to the tectum are not fully understood, it is thought that ATO7 and other downstream transcription factors can modulate response to secreted molecular guidance cues. What is better understood, however, is the interplay between different cell-bound molecular signaling mechanisms that determines the final position of axons in the tectum. In the retina, EPHA has a nasotemporal graded expression and EPHB has a dorsal ventral graded expression. The EPH receptor expressing axons that enter the tectum are first attracted by a low concentration of a corresponding ephrin ligand, but are also repelled by higher concentrations of ephrin. Put simply, axons of retinal ganglion cells that express a higher concentration of an EPH receptor will seek a location in the tectum with a lower concentration of a corresponding ephrin ligand, and vice versa. It is well known that the relationship between EPH receptors and ephrin are important for fine-tuned positioning because knockouts of both EPH and ephrin will lead to unrefined retinotopic maps. At this point, you have hopefully noticed a trend. Coarse map formation is often directed via secreted molecules, and finer details are often controlled via cell-bound signaling. You might have also noticed an emphasis on the interactions between EPH receptors and ephrin for cell-bound signaling. This weighted importance on EPH and ephrin has even been seen in sensory modalities whose map formation is still not quite understood. For example, the development of the somatosensory and tonotopic maps are still not well understood, and even though all the molecular signals important to this development have not quite been identified yet, it is known that EPH and ephrin still play an important role. When ephrin and EPH are knocked out, inputs into the topographic map become unrefined. In order to understand how these maps are affected, we must also gain an understanding of how these maps reflect information. The somatosensory map reflects information relating to sensory distribution, density, and activity. This mapping is best visualized when dermatomes are mapped onto the cortex. When ephrin and EPH are knocked out, thalamic inputs into the somatosensory cortex become unrefined. Although not quite proven, it also seems to be a reasonable guess that axons from the thalamus are guided into the somatosensory cortex via secreted molecular signals in a manner similar to what has been seen with the olfactory map development. The tonotopic map, on the other hand, is a mapping of frequencies to the brain. The cochlea actually encodes a unidimensional reading of frequency, and as such, the map is actually a direct mapping into the primary auditory cortex. Preliminary studies in mice have actually shown that when ephrin A3 is knocked out in mice, the tonotopic map becomes significantly unrefined, resulting in blurring between frequencies within the anteroventral cochlear nucleus. And although these knockout mice are still able to hear, they exhibit impaired frequency discrimination. Hopefully, through these sensory map development examples, you were able to see and understand the different use cases for secreted versus cell-bound molecular signaling. Although understanding these development processes and the signals that underlie them is still not fully understood, we can utilize information from processes that we do understand to use them as a starting point for deeper exploration into learning about map formation.